It is amazing. I don't know how many of y'all are involved in irrigation, but we're involved in this Ogallala project. And, you know, these irrigation companies and researchers and, you know, consultants, they're working hard to figure out how to automate these systems. And just like Brian can tell you with precision fertility and seeding rates and all that stuff, the technology is probably, their capacity to do things is probably beyond their scientific understanding of why to do them. But it's rapidly developing, and the canopy temperature stuff that he's working on is part of that. So I'm going to walk over here, talk to Cameron. I'm going to talk a little bit about soil health testing procedures. In my prior life as a PhD student at Virginia Tech, I was actually more of a chemist than I was a conservationist. And so I had a lot of experience starting here at OSU with Lynn and some of the other soil scientists learning about laboratory procedures and things like that I don't always get into anymore. But I wanted to spend some time on this. The NRCS, they released their recommendations for soil health indicator assessment, and so we're going to run through some of these things and then look at a little data on all of them, or on some of them at least. And so, and rightly so, they name, you know, some of the characteristics that make a good soil health indicator. Same thing goes for, you know, other analytical protocols we do for soil analysis, whether it's nutrients or pH or salinity. But they need to be effective variables that are sensitive to our management strategies, whether it's looking at no-till versus conventional till, or no-till rotations versus monocultures and things like that. The idea is to get at numbers that we can look at the soil and see where we're at and understand how that management is affecting it. They want variables that can be detected, changes can be detected in 1 to 3 years, and it represents soil processes or conditions relevant to agriculture or the environment, like what Nathan was talking about. So we want analytical procedures that produce numbers that are relevant to our agricultural practices or environmental outcomes, that are sensitive to our management such that we can see changes over time. Another one, and a big one for me, is it ready for commercial laboratories? You know, in a research laboratory, I can spend a lot of money analyzing the soil, and we do. We've got cheap labor and students that we can threaten with lack of graduation to get them in there working for pretty much free. But when we go into a commercial lab, we can't do that. So we have to have, first, protocols that you guys as ag professionals in the field can use to collect those samples that aren't ridiculously complicated. And then we need cost-effective analytical procedures in the lab per sample. And then specialized equipment and space needs need to be limited. You know, and all that kind of depends on how much my throughput is. I can afford a $500,000 machine if I can put 1 million samples through it every year. And so those kinds of things have to be balanced when we look at these. Another one that's big for me is the reproducibility of it. You know, I had a soil chemistry professor. His name was on a lot of books. He's freaking awesome. And he was educated by people whose names were even on more books, that are older than me by 3 times. And these guys, they taught me that error and limiting error is important. Our ability to reproduce a number from Oklahoma State University to Ward Labs to JNL to all these different labs, these numbers need to be reproducible and they need to mean something. And on top of that, they need to be able to measure a difference. So if my difference is 100%, but my variance is 200%, then my difference is 0. Does that make sense? And what I said is if my difference, if I had 100% difference, meaning I have 50 and I have 100, that's 100% difference, but my variance on each number is from 0 to 200, then that's a problem. And that's where we get reproducibility. We need to be able to reproduce that number over and over from lab to lab. And then interpretable for management decisions. This is where I think we're working on pretty hard, is the values need to be understood. Management to affect change in those indicators need to be known. And then regional potential ranges for indicators need to be known. And then and this one's an interesting one too, in a state where you have 50 to 15 inches of rainfall with soils that are this deep versus this deep, made of clay loam, silt loam, sandy loams, and whatnot. And so those regional ranges need to be understood just like we have an understanding of our organic matter content, our pH ranges, like what we get out of Highland's talks when he talks about the state of soil fertility in Oklahoma. And then thresholds are known. We need to know how will yield, resilience, risk, or environmental outcomes be impacted with these numbers. And this is probably, and Nathan, you know, he discussed some interesting things. We have a lot of discussion about soil health and the environmental benefits, just like we have a lot of discussions about, you know, precision nutrient management and the environmental benefits. But the data is pretty thin. We expect it to be beneficial, but the data can sometimes be thin when we talk about some of these things. And so we've got a lot of work to do in this soil health assessment realm. I'm going to start off with organic matter. It was the first one on the list of recommended indices. It's very well understood, very seldom analyzed in routine soil analysis, but it's very well understood. We accumulate at the surface of stable soils. We lose it when we flip dirt under cultivation. If we clear trees, if we clear native pastures, we're going to lose organic carbon. Adding material with high seed-in ratios will typically increase our organic matter. Intensifying our cropping systems will intensify or increase organic matter. Growing cover crops with high seed-in ratios, like I say, will increase that organic matter. It can decrease nitrogen availability. As you have a lot of straw out there. Sometimes if you don't have enough nitrogen, you can have yield-limiting conditions. Typically, we can see an increase in soil biology, and structural character would be a benefit, and the function of soil productivity and clay content can affect it, though. This right here is where you get variance. You know, you can see a soil up on the Vertigus River that may have 6% organic matter, and then down here in Perkins it may have 0.75% organic matter, and they'd be under the exact same management. And there's a struggle. So you have to have that context in the soil. Inherent soil characteristics influence that a lot, as it does in most of these. Analytic capacity is very easy. We can run these samples. You can run lots of samples for organic matter through Highland's lab. They're very well established, highly reproducible, and very consistent numbers. One of the older ones was Walkley Black. It uses dichromate, so we don't use it anymore because that'll cause cancer, and we don't like disposing of that with our health and safety people. So we use dry combustion. So this machine drops that sample down into 1200 degrees Celsius furnace, ignites it, sends it through a gas analyzer, tells us how much CO2 is in there, and we get a number. Then the loss on ignition is one that you can do if you've got a 1000-degree furnace and you just weigh the sample before you put it in the furnace, weigh it after you put it out of the furnace, and you determine the weight of the carbon lost. And so organic matter is a good one. And I was at a meeting a few years ago, and we were talking about soil health indicators. It's fascinating because some farmers, came to the conclusion that if we were going to want to look at soil health, the first thing we need to do is really start talking to people about organic matter. How many of y'all have organic matter analyzed on your soil samples when you send them to soil testing? It's a pretty good number. That's better than average, so you guys are better than average, I think. But that's the first thing, because you can get an idea what's going on with your soil, and it's a very routine sample analysis. So structural stability, that's what we're talking about, aggregate stability or soil structure. It's a sensitive disturbance in organic matter content. Is also a function of clay content. So if you have a sandy soil, it's not going to have much structure. A loamy soil is going to have a little more structure, and then a high clay content soil will have structure, but it may not be very stable. And that's where you get into slaking and dispersion and things like that. And that's why stable aggregates are important, because they promote porosity, and porosity, it has everything to do with effective water movement and air movement and root movement in the soil. And so that the stability of those aggregates is very important, and the size and character are important. Stable aggregates limit the effect of, like I say, dispersion and slaking. And the good thing about this one is there is significant data in the literature to understand current method thresholds. So when we look at stable aggregates versus unstable aggregates, we understand how that's going to affect erosion by water and wind. My friend Deanne Presley, she loves doing all this work with, she's up at K-State. Another good reason why I like to have K-Staters down here, because they really like to work hard, much more than I do. But Deanne, she does a lot of this stable aggregation stuff, and it's a lot of freaking work. And the reason it is because you take soil and you put it on a nest of sieves. First you have to kind of dry it and then softly grind it, put it on the sieves, and then you dump those sieves into a water bath of some sort, and you shake it in a water bath for 10 minutes. Then you take the sieves off, you wash the soil off the sieves into a pan, you dry it and you weigh it, and then you put them back on the sieves, and then you agitate them and break them apart to wash all the clay and silt off of it, so you can determine how much rock or sand is in that sample, because you have to subtract that from the number for some reason. And long and short, it's like 4 days worth of work to run these samples, and that's why I don't do it, because I'm lazy. But anyway, you break up the aggregates and sieve them, and like I say, 1 day is for initial sieving, 1 day is for drying and washing the sand, and then 1 day is to weigh the sand and calculate the results. And so this is how research guys do it. This is the method proposed under the current NRCS guidelines, and it's going to be very expensive. Although if you want to send samples to New York at Cornell, they'll do it for $20, and they're losing lots of money on that deal, I can guarantee you. But $20 for that is cheap. But the challenge is it's very little opportunity for automation. It's very labor intensive. Analytical characteristics required, or this line here, I meant to say, the analytical characteristics of the method require patience and or oversight. You can't just hire a $10 an hour kid to do this, because he'll blow his brains out and then it'll mess up the results. And so I'm serious, I don't know how people do it. There's a lot of work, but most likely very expensive as it goes into commercial use. There are some single sieve methods that we're looking at, again, because I'm lazy and my students are lazy too. But there are some single sieve methods that give you estimates of slaking, which is really what you're looking at for stability of aggregates. That multi-sieve method gives you the distribution of the size of aggregates. But if you're really only interested in slaking and dispersion, then we can use simple single sieve methods. Might be a little cheaper. Then we go on to microbial activity. So that's your CO2 burst. Dr. Rick Haney was a big fan of that. And it was really being driven there for a while. You don't hear about it so much anymore, but you'll hear about this Slovita method, and then the Haney soil health assessment tool is pretty much driven on that with respect to soil health, and he's got some nutrient analysis in there. But the Slovita, it just gives you CO2 emitted after a 24-hour period, and it's a pretty good method in general. Now we're looking at a 4-day method. I'm not sure why, because if you talk to Ray Ward, he's not going to like 4 days. He's going to like 1 day turnaround because he's moving product through his lab, and 4 days is going to be a challenge for him. But I think in the grand scheme of things, that 4-day method is probably going to be highly correlated to a 1-day method. We have done enough work in Oklahoma in my lab to know that CO2 burst is more sensitive to management than the organic matter, and so you get a little bit more information. The methods are typically, like I say, highly correlated, but they're extremely sensitive to the drying condition that you put the soil under, to the temperature of the lab, to the hours under incubation. There's a lot of things that they're very sensitive to. And just here's an example. This is the Slovita method down here, and this is a method that we use in our lab because it's a lot cheaper for us. But you see a very high correlation. But he tends to get saturated about 180 part per million. So now he's going to use bigger jars and smaller soil sizes. And when you send samples that you may have sent 5 years ago to a lab that runs Slovita, you're going to get different numbers because he's changing his method. And so be aware of that. As we proceed, we're going to get a lot of changes in the outcome of these methods if these guys that are putting them forward don't pay attention the absolute change in values they get. And this is just an example. So this is Selvita, and this is this gas chromatograph-based headspace analysis that we use. You can see his numbers are about 3 orders of magnitude or 3 times higher than ours, and then they all increase with increasing drying temperature from air dried to 45 degrees C to 65 degrees C, and then they crash at 105 degrees. And so you're increasing that CO2 emission for both methods, and then the difference between the conventional till and the not tilled treatments, it decreases, right? So when it's air dried, you have a much larger difference between 125 versus 50, and then when you get up here to higher temperatures, you get 162 versus 119. That is the effect of that saturation that is occurring right here, because he's getting more and more numbers that are 180. And then here our numbers are smaller, but you have 50 versus 24, so you got 112% difference. But not only is that difference important, but then this average coefficient of variation is important. As you dry a sample at higher temperatures, the variance goes down. And there's some reasons why we won't get into. But that chemist that was a mentor of mine at Virginia Tech, he talked about soils as being reagent grade dirt once we dry, grind, and sieve them. And that's what we want in reproducing numbers and reducing that CV, is we don't necessarily have to recreate the field condition. We just need to have numbers that are meaningful, that can be reproduced, and we can do that with reagent grade dirt. Then carbon food source is another one that people look at. It's the easily oxidizable organic carbon is one method. You can look at particulate organic matter. You can look at 28-day carbon mineralization, which is a CO2 evolution method. You can look at a cold and hot water extraction of organic carbon, or you can look at soluble carbons. And these are just a list of the things we could analyze that you could get a carbon food source number out of. And the current, the current method of choice is this easily oxidizable organic carbon or permanganate oxidized organic matter. And permanganate's oxidizing agent that turns color depending on the oxidative state of the solution. So it's a real simple deal. It's a simple wet chemistry analysis followed by just a colorimetric analysis of the color of that liquid. And the procedures where, like I say, permanganate is oxidized, where they oxidize the organic carbon and the solution changed colors, and that's why they like it, because it's cheap and simple. Here's some data from some of the work that one of my graduate students did, is the CO2 burst versus active carbon. You have a fairly good R square, but the important thing is you have some of these outliers, and those outliers are those points where you might be learning something. And then organic carbon, those same samples are outlined against the relationship between organic carbon and active carbon, but they're at the other end of or on the other side of that regression line. And so active carbon does tell us, I guess the moral of that story on that data is that active carbon tells us something different than organic carbon to some extent, and it also tells us something different than CO2 burst. The question is what does it tell us? And from an agronomic standpoint, when we ran any of these methods on any of our cover crops or long-term versus short-term no-till plots, we really didn't get much of a difference. And so these aren't sufficiently sensitive to pick up some of the differences that we're trying to observe in our management plots. And then you have this available nitrogen, bioavailable nitrogen. So that's different than ammonium and nitrate. This is thought to be like organic and inorganic nitrogen that can be extracted to get a little more information on top of like an ammonium nitrate analysis. And the way they do that is autoclaved citrate extractable protein content. You can also do it. Haney used a cold water extract, just water-soluble organic nitrogen. You have 7-day anaerobic potential mineralization of nitrogen. Illinois soil nitrate test, and we've worked with that and not seen much of useful information come out of it. And then you have glucosaminidase activity. And I just put all those on there so that you understand the diversity of these different methods where all these guys that are looking at these protocols are looking. But I'm going to stick to the ones that are highlighted in that NRCS guideline, and the one they've highlighted is that autoclaved citric acid extractable protein. And they say it's an indicator of the fraction of the soil organic matter pool that is present as proteins or protein-like substances, and then very well associated with overall soil health status because it represents the largest pool of organically bound nitrogen in the soil. And that's coming out of the soil health testing program at Cornell. So I found this data. This is data from our friend Deanne Osmond out of North Carolina. She's kind of the, she's a crossbreed between me and Hyland and Brian, I think. Is that right, Brian? Yeah. She's extension soil specialist, does a lot of soil testing. But they had these 20 to 30-year-old long-term systems research plots. And I've highlighted two of them. Some of these plots are 30 years old in here, and one of them was a coastal plain, one was a Piedmont. They had no-till, chemical-based, management of weeds and whatnot, and they had conventional tillage, and they had conventional tillage organic system 1 and organic system 2. Then they had no-till versus strip till versus spring disc versus fall chisel, spring chisel, a fall chisel and disc, a spring chisel and disc, a fall moldboard, and a spring moldboard. So they had a lot of nice tillage management strategies in here. And they looked at some of these Cornell soil assessment tools, and one of them is aggregate stability. And what you'll notice is there are no A's and B's along that line. That means there's no statistical differences. They looked at organic matter, and they found no A's and B's, meaning there's no statistical differences in any of those numbers. And so I want to point out again, there's a 39 versus a 27 in this study, or as little as a 22, and those numbers are not statistically different. Their magnitudes, the relative difference between the two numbers, pretty substantial, 40 versus 22. So the no-till has nearly twice as much organic matter in it, but it's not statistically different because of the variance and that error. And then you start looking, another one that wasn't statistically different is this active carbon. So they did find significant differences among treatments at Piedmont for this protein extraction and that 4-day CO2, and then they had differences in corn and soybean yields. Now these were point samples collected at a time in the spring, and then these yields are average yields over the 30 to 20 years that these studies were conducted. And so we just look at this soil protein versus soybean yield, and what you'll see is there's not much of a relationship, an R squared of about 0.5, but then on the corn you have a relationship across both locations with an R squared about 0.77, and that's pretty good. And so what we're seeing is that protein value is related to the historic corn yield, and most likely because those soil systems with high protein at this point in time are related to systems with elevated nitrogen in them over that period of 20 to 30 years. And then when we look at CO2 burst, here's the corn yield. I flipped it around on you to kind of keep it interesting. But this is corn yield. And now with CO2 burst, there's not much of a relationship with corn yield. At R squared about 0.4. But in contrast, the soybean yields over here, it has an R squared of about 0.77. And so that suggests that microbial activity, the residue, the organic matter, is more influential in affecting yield for soybeans than it is corn. Over here, that corn, if you have nitrogen limited conditions with high carbon content that maintain a better baseline CO2 level, then you're not going to have much relationship because there's too much going on there with nitrogen. Another thing about this CO2 burst is if you notice right here, this 32 and that 33, they're the highest numbers on that board, and they're both from spring tillage activities. So that tells us that there's a lot of history we need to know, both multi-year prior history as well as recent history on soil management if you're wanting to interpret that. But they are showing some interesting data as far as some of these outcomes and how some of these variables might be different with respect to relationship with yield on a legume versus a grass. And then here, this is organic matter. So it's absolutely useless number to understand organic matter versus historic grain corn yield. But then on the soybeans, the higher the organic matter, the better our soybeans yield with an R-squared of about 0.6, which is not bad for this kind of data. And so those are just examples of when we start mining data and looking at it real close, we can see some interesting things. I'll finish with this method, this phospholipid fatty acid deal. People have been talking that for about 5 years. I still don't know exactly what kind of information we're going to get out of it, but essentially they're going to tell you what the biological diversity of the soil is. So they'll tell you the G, or the gram-negative, gram-positive bacteria, anaerobic bacteria, actinomyces, methanotrophs, single-celled prokaryotes, eukaryotes, fungi, avascular mycorrhiza, and these are all things that you hear about when we talk about soil health. And I'm not saying they're not important. I'm just saying that our capacity to understand how all these things interact is very limited, in my opinion. And this is just some a little verbiage off a soil health assessment from a lab that will go unnamed. But when you just look at something like the fungal to bacterial ratio, it says bacteria tend to dominate in systems with few organic inputs. And then down here, it said dry conditions, slightly alkaline, 2 alkaline pHs, or increased land disturbance through prolonged and extensive tillage, grazing, or compaction may also favor bacteria. And I point those out because, you know, fungi is going to be more prevalent in a stable, high organic matter type condition, but then soil dryness or wetness, soil pH, a lot of different things are going to affect that ratio. And that's one of the challenges with that, is that we like to understand the diversity of the soil biology, but then it's extremely difficult to interpret with a blind eye. So you have to know a lot about the history of that field and some of the other variables within that soil to understand that number. And that's where we're challenged on these soil health indexes. But I'll leave you, I like to show this. This is where I'm after. I've had, 5, 6, 7 years ago, I had discussions with our NRCS conservationists about how we could quantify that. And so I'm still interested in working towards it. And that's what this soil health assessment program that's really going across the nation is working on. Because if we can figure out how to better quantify what we see in this dark organic or soil with high organic matter, then we can start making grain a little bit better, because it does have impact on our crop performance. But the challenge is that it's very complicated. Efforts are on their way to provide assessment tools. We're a long way from a meaningful index, and by an index I mean a single number that tells you how healthy the soil is. And the reason we're a long way from that is that healthy soil as defined is one that's productive, that has limited environmental impacts, and all these things. So we're asking a lot of this index, to be honest, and we're asking it to be, universal. And what we find from the North Carolina data is that if you look at just their index, when you combine all these variables from soil analysis, it's not useful in assessing yield. But if you look at some of these individual components, they can be related to yield. So by that I mean a soil health index as one number from a combination of other numbers is going to be a challenge because of the complexity of that question. But with context, you can look at some of these specific variables and start to understand productivity. One another thing is that we're always going to need some context with respect to the sample time, whether spring or fall or winter or summer, the cropping history, the fertility, soil type, etc., to understand some of these variables that are so very dynamic, particularly the biological ones. The usefulness of the indicator in assessing productivity, like I say, will be dependent on crop type. Usefulness of the indicator in assessing environmental impact has had little attention, in my opinion, just like Nathan said. There's a lot of work to be done on environmental impact assessment and how they interact with some of these variables. And there is a great deal of standardization needed. That's one of the things that's aggravating with me. I don't care what tool we use as long as it's standardized and it can be used across the state. And so many analysis are very sensitive to protocols that are used, and then the numbers will change as methods are refined. And so if you're looking at some of these methods in your program, don't be surprised if the numbers suddenly change. And that's based on my discussions with some of my friends up at Kansas, where they send them to a commercial lab and getting a host of things analyzed, and then they'll send another set and the data is completely different. And that's because they're refining the analytical reproducibility and capacity of the method, and in doing so, they tweak things like the jar size or the incubation time or this, whatever, and it will grossly change these things, and so we need to be aware of that. And with that, I'll take any questions. I want to put a plug in for this all crops school as well as this irrigation conference there on the 28th of October or of February there in Goodwell.